Dr. Quratulane Gillani, and I am an assistant professor in Women University. The presentation which I'm going to deliver today is anti-obesity potential, toxicological evaluation of Papaver Somniferum use in obese rat models. As is generally known that obesity is itself a disease, and we define the obesity as a person whose body fat percentage is dangerously high and does not reach the BMI less than 20. Obesity can cause so many other diseases like heart diseases, increase the risk of heart diseases, blood pressure, diabetes, cancers, and it's also very dangerous for your health, mobility. It's reduce the mobility of the human beings. Obesity can cause by three different effects. One is the genetic effects. We have some genes which can control our obesity, and due to which obesity can cause in families, and some have the abnormal hormonal responses. These hormones cause problems to body physiology and increase the body weight and cause obesity. And third form is the obesity is due to the high consumption of the junk food or high fat diet intake. High fat diet cause so many problems. It clogs your arteries. It can increase the your heart problems. It increase the blood pressure. In other terms, if obesity can cause all these problems. Now, the medicines which are available in the market are normally very high cost, and they have a very dangerous health problems or side effects. There are many plants or herbs which are used medicinally to reduce the obesity, and journals generally in many medical pharmacological companies use these plants. The one plant which I'm talking about is the poppy seed plant or poppy plant, Papaver somniferum. This plant is used for the collection of the opium, and the seeds of these plants called the poppy seeds or the hashhash. Hashhash is very common in the households, used in many recipes. It also has a very alpha-linoleic acid in it, which can help in the reducing the obesity. It's omega-3 fatty acid, and it can be helpful to reduce the obesity. In our research, the main objective of our research was to see the efficacy of the Papaver somniferum against the obesity, and check the toxicological effect on the liver or kidney. If it is useful, it is safe to use as a obesity for the obesity. So in the first trial, we induced the obesity in the animals. So our first part of the experiment, in which we divide the group, one is the control, which have the normal diet, and the second with the high fat diet, in which we feed the animal with the breads, with the high fat and animal fats, and all those protein fats which can cause the obesity to the animal. And it's the feed was prepared with the percentages of these fats, and then we give this feed to the animal for the 6 weeks and induce the obesity. Then we divide these groups, this fat group, into further 4 groups, and then one became the control positive, in which have obesity, but we didn't give any type of treatment to the animal. In standard group, we give the animal the medicine which are available commercially in the market, and the third and fourth group was treated with the poppy seeds. But in these 2 groups, difference was that one is washed poppy seeds and other was unwashed. Because poppy seeds has opium on it, and when we washed it, it's wash away the opium layer on the poppy seeds. So after the dose was which we give is the 30 gram of feeds, 0.5 gram per 30 gram of the feed we gave on the daily basis to the animals. After that, we dissect the animal and collect the blood, and we did the experiments like we did lipid profiling, blood glucose test, which we did throughout the experiment, and the liver function test, kidney function test, CBCs, and also we did the histology of the animal with the kidney, liver, and the obese. Um, then we did the statistical analysis, two extra studies which we performed side by side of this experiment. We use this effect, the poppy seeds on the saliva digestion. Saliva has the enzyme called amylase which digests the carbohydrates. So we use this amylase, and we see that if poppy seeds reduce the digestion or did not have an effect on the digestion of the carbohydrates. And second was the behavior study. In first group, the result, the obesity induction trial, in which we divide the group two into and give a normal diet to this control group, and um, high fat diet to this group. And as you can see in this graph, that weight is definitely increasing due to the obesity weight gain, and it is statistically very significant result. In second group, we divide these animals and into 5 groups, and then we give poppy seeds standard medicines to 3 groups, and the 2 was our control negative and control positive for 4 weeks. It shows that unwashed poppy seeds reduce a little bit weight in week 4. So there is no very highly significant result in other groups, but it still have some effect on the weight, very low effect on the weight reduction. If we see that the effect of the poppy seed on the glucose level during the experiment, uh, we monitor the glucose very carefully. As you can see in this graph, the value is highly statistically significant, and that the washed and unwashed animals have very low glucose level as compared to other groups. Again, with the HDL levels, which is high density lipoproteins, these 3 groups have low levels as compared to these 2 control groups. Again, in the low density lipoproteins, again the result was same and significant, that it's reducing the low density lipoproteins, and also it's reducing the very low density lipoproteins, and this value is very highly significant. Again, there is no very prominent effect of poppy seeds on the liver functioning or kidney functioning. Overall liver function or kidney function of the animals show the same results, and the damage of the kidney or liver is due to the obesity which we caused by giving the animal high fat diet. So there is no other effect on the animal which are given with the poppy seeds. The other experiment which we did on the side wave, in which we study the washed and unwashed poppy seeds effects on the digestion of the carbohydrates. And in this experiment, you can see this A test tube have a glucose and iodine. In B, have a glucose solution with amylase, and in C, we have amylase, glucose solution, and poppy seed solution. So you can see it's slowing down the digestion. It's not completely digested, This picture is taken after 15 minutes. This picture is taken after 30 minutes, then 45 minutes, then 1 hour, and then 1 day. And all these pictures show that it's again reducing the time of digestion of the carbohydrate by amylase. In this pictures, these are the washed poppy seeds, and again these shows a slow digestion of the glucose by the amylase. That's mean poppy seed and in the washed form are more effective for the carbohydrate digestion inhibition. So it's inhibiting the digestion of carbohydrates, which is a very common factor in the high fat diet, and it's increasing weight because carbohydrates are more generally used as energy production and production for the energy production. So it accumulate more easily. The other group test in which we did the rota-rot test to see the mobility of the animal, and it's very obvious result that control group, which is not obese, have a very high, very effective on rota-rot, very mobile, and obese and treated group is not very mobile in this experiment. And in the open field also, the mobility rate is low in the obese animals, but it is high in the control group and the group which is treated with the standard medicines for the obesity. The other experiment, which was the comparison of the noble object recognition, the familiar objects, the results show not very significant effect on any of the groups, and with the unfamiliar objects, there is not very statistical difference between the other objects also. So it means there is no particular effect on the learning behavior of the animal. So in the conclusion of this experiment, we can say that poppy seed help in reducing blood glucose, HDL, LDL, VLDL, and obese rats, which are on high fat diets. Poppy seeds did not exhibit any adverse effects on the health of the obese rats, and poppy seeds slow down the digestion of the carbohydrate by amylase enzymes. So there is no adverse effects of the experiment on the learning behavior on the animal. So it is very useful that we can use these poppy seeds for the obesity reduction because the percentage we use, the concentration we use, amount 0.5 gram is very small. So in future we can use a high dose of this poppy seeds to see the effect on the obesity, or we can produce extracts of the poppy seeds, and we can use oil of the poppy seeds, and we can see what kind of effects they can cause on the obesity. Thank you very much for your time.